Welcome. I'm Andrew Hunter, Director of the Defense Industrial Initiatives Group at CSIS. Thank you so much for joining us today. I want to briefly introduce the third event in our series on synthetic biology to set the frame for today's discussion with our experts. Our first event in this series in February focused on the present and future promise of synthetic biology to the economy and to national security, with Dr. Diane DeEulis, Dr. Jason Kelly, and Dr. Gigi Gronval. In our second event last week, we focused in on national security, hearing from Dr. Tara O'Toole, Executive Vice President at InQtel and former Undersecretary of Homeland Security for Science and Technology. Today's panel is the second part of our look at national security, and will revisit in depth the issues discussed by Dr. O'Toole on the risks and opportunities of synthetic biology. Now, this topic couldn't be more relevant to current events, given the way that COVID-19 has upended our daily lives and disrupted the global economy. If you missed either of our two earlier events, you can access recordings of them on our website and on the CSIS YouTube channel. The series will continue in coming weeks with greater examination of the economic potential of synthetic biology. I'd particularly like to thank Ginkgo Bioworks for providing the support that's made this series possible. Now let me turn things over to Dr. Morgan Dwyer, the Deputy Director of the Defense Industrial Initiatives Group for Policy Analysis, who's leading our series on synthetic biology and will moderate today's discussion.
Thanks, Andrew. So I'm really happy to be continuing the discussion that we started last week with Dr. O'Toole. For those of you who tuned in last week and submitted questions, if we didn't get to your questions last week, I look forward to getting to them today. And also a reminder for everyone else who is tuned in, you can submit questions for our panel online, and we'll get to them later in the session. So before I introduce our panelists, I wanted to start off by just reminding everybody what synthetic biology actually is. So synthetic biology is a field that employs engineering approaches to modify or create biological organisms. So essentially, we can think of biology as code that we can read, write, and edit. And there are lots of applications of synthetic biology that I hope to explore with our panel today. So for that panel, we have three experts. I'd like to start off by introducing Dr. Rocco Casagrande, who is the founder and managing director of Griffin Scientific. We also have Dr. Megan Palmer, who until recently was a senior research scholar at the Center for International Security and Cooperation at Stanford, where she remains an affiliate. She recently assumed a new role as the executive director of leadership initiatives as part of Stanford's Department of Bioengineering. And finally, we have Dr. Alexander Titus, who also recently assumed a new role as the Chief Strategy Officer at the Advanced Regenerative Manufacturing Institute. Before that, Alexander was the Assistant Director for Biotechnology at the Office of the Secretary of Defense at the Pentagon. So I want to welcome all of our panelists. I'm really looking forward to the discussion today. And with that, I just want to dive right in with some questions. And I want to start off by talking about what risk means in the context of both synthetic biology and national security. So Megan, I'd like to start off with a general discussion of dual-use technology. So synthetic biology is sort of a classic dual-use technology. So could you help us help our audience understand what types of national security risks dual-use technologies pose, and if any of those risks are unique to synthetic biology?
Sure. Thanks very much for having me today. So as you said, biology poses a sort of classic dual-use problem, and dual-use risks really relate to research with legitimate scientific purposes, where the results, both the knowledge and the products, can be misused to pose threats to public health and to national security. And so arguably most, if not all, of biotechnology has dual-use potential. And dual-use research of concern has been used to represent that subset that is the greatest threat to security. And defining exactly what areas are of concern is a pretty long-standing debate and is still far from a science, and is recently focused around concerns around research with potential pandemic pathogens, although it's certainly not limited to this. The debate has relevance beyond biotech, but there are unique facets of of biotechnology that make this especially challenging. Biology is a powerful distributed technology that works, and it has vast potential for public good, including protecting us from the organisms that are trying to kill us all the time. And we are made from biology, so it makes us especially vulnerable. And so in this case, we need to make sure that as we manipulate life, our capacity to manage these types of dual-use risks are advancing just as quickly. And lastly, just in synthetic biology, one of the unique facets is that we are both recombining and synthesizing new functions, and these new functions are within organisms that can replicate. So it poses a rapidly evolving challenge of how to manage these risks.
Wonderful, thank you. So I want to build off that question a bit. You highlighted the fact that there are researchers focused on synthetic biology, and presumably a lot of that research takes place in laboratories. And so I wanted to ask you, Rocco, to talk a little bit about lab safety in the context of synthetic biology, and particularly help us understand are the risks that synthetic biology poses to laboratory safety protocols, have they evolved with the technology, or are they the sort of traditional risks that all dual-use technologies pose?
Well, that's a great question. I think one of the things that's important to note is that the vast, vast majority of synthetic biology research has nothing to do with pathogens. Most of the more interesting functions relate to the ability to create new molecules or new materials, or create existing materials in new, more efficient, more green ways. That being said, one of the risks that the massive parallelism that you can see within synthetic biology poses is that you can try out more of the biochemical space, given some of the more advanced tools of synthetic biology, and explore combinations of proteins and nucleic acids that have never been experienced in nature. In doing so, it's very difficult to assess risk. We don't know exactly what the potential of those pieces are. But that being said, because pathogenicity is such a highly evolved phenotype, it involves the coevolution of the pathogen with its host. It's really unlikely for new pathogenic traits or new pathogenic strategies to just completely happen by chance. So overall, the biosafety risk of synthetic biology as a field is low. Now, that being said, people are trying to apply tools of synthetic biology to really understand more about the host-pathogen relationship and understand what it is that makes pathogens dangerous. In that context, the power afforded by synthetic biology could lead to the creation of pathogens which have enhanced ability to cause harm or transmit. The fact that we can explore more of the biochemical space than we could previously kind of puts a lot of challenges to risk assessment and control that we previously relied on to keep ourselves safe. So if we don't completely understand all the possible ways that a protein could become a toxin, let's say, it's impossible to write a list of what all those ways are down to say, okay, you only can work with proteins that look like X, Y, and Z under these levels of containment, or you can only ship proteins that look like X, Y, and Z to unknown customers or to countries of concern, potentially. Additionally, I think a lot of the promise of synthetic biology lies in our ability to grow life forms out in the environment, whether they are new crops or, let's say, algae produced in contained reservoirs, or even plants that could change the environment for good, like bioremediate heavy metals in soil. I think there's an additional risk in synthetic biology that because these are self-replicating organisms, the introduction of engineered organisms into the environment for a variety of beneficial purposes could get out of hand, one, but also those same tools that teach someone how to release and how to adapt an organism to its environment such that it can be sustained in the environment could be used by those outside of traditional institutions that might adhere to the norms and regulations that relate to environmental release. And you could have people with less awareness of these norms releasing organisms that they probably shouldn't after leveraging what was learned in academia, for instance.
Wonderful. Thank you. And that helps me pivot to the question I was going to ask Alexander, which was really focused on sort of the broader applications of synthetic biology that Rocco highlighted. Alexander, your role previously at DoD was really focused on shaping DoD's biotech investment strategy across that entire biotech space. So I wanted to ask about the question of risk from a slightly different perspective to you. What's the risk if DoD or other agencies in the U.S. government do not sufficiently invest in some of the biotechnologies that Rocco highlighted in his response?
Yeah, absolutely. Thanks, Morgan. That's a great question. And to build off, a perfect build off of what Megan and Rocco were both talking about. So if you think about everything we do needs to take into account all of the risk considerations we were just talking about. But if we slightly reframe the perspective to biotechnology being an opportunity space, and one of the opportunities is to mitigate a lot of the risks that we're concerned about, COVID-19, then it starts to give us our ability to think about opportunities in other areas as well. And if you think about investment from the federal government's perspective, the federal government is established to represent and protect and help Americans thrive. The U.S. federal government help Americans thrive. And so part of that, a huge part of that, is economy. So if you think about all of the applications that the federal government could develop for specific mission sets, whether it's defense or energy or health, those then have major implications into the broader economy. And what biotechnology really is giving us is the ability to have an entirely new vector or dimension of technology development. So if you think about it, for thousands of years, we've had physical solutions to technology needs. So I need to move a heavy object from point A to point B, so I push it, pull it, or roll it. Then in the Industrial Revolution, we had synthetic chemistry, and we had an entirely new dimension of chemical development, which gave us new technologies that changed the world. And so now as biotechnology becomes the third, and you have a physical, chemical, and biological component to technology development, or some combination of that, to what Rocco was saying, we get to explore chemistry spaces that are previously not reachable through traditional synthetic chemistry means, which means our ability to get to new materials, which could be lighter or more environmentally friendly or stronger or a combination of all of those, could have major impacts on everything from sports equipment to military uniforms to the kind of material we build car seats out of, for example. And so the opportunity and the economic potential of what we call the bioeconomy is enormous, and it's only growing. So we've had pharmaceutical companies and drug companies, biotechnology companies for a long time, but now we're being able to expand outside of just human health to other industries as well. And I'm sure we'll talk about that over the next 45 minutes or so, all the different application spaces for where biotechnology can impact. But this is all to say that most of the technologies that really start to grow from a nascent field into a booming economy have some or large investments from the federal government. There's a lot of great examples of technology that came out of, say, DARPA, the Defense Advanced Research Project Agency, Siri, the internet, these kind of technologies were seeded with federal funding, but then grew into major applications. And so the risk, COVID-19 is also showing a major issue where economies are affected on a global scale when we have these pandemics, right? Everyone, myself included, looks at your retirement account and goes, oh man, that's crazy. But those economies then can be, one, protected and also enhanced by going into these new areas of technology development. And so that's really what a lot of the promise is. And then if you also think about, from the context of national security, economic security and economic strength is fundamental to national security. And so the countries with the strongest economies generally have the strongest national security base as well. So it's an interplay that takes it beyond the traditional kind of missiles and rockets and those kind of things, and when you think of national security, to a much broader and more strategic opportunity and risk space.
Wonderful. Thank you. So I think all of you started pivoting in your answers to how do we govern these risks, which is exactly where I wanted to go with the next set of questions for everybody. So building off of Alexander's discussion of what the larger bioeconomy is, Megan, I'd be interested if you could help us understand who are the key organizations within that larger bioeconomy, be it private companies or governments or researchers or bad actors, and what governance structures exist to control the relationships between them. And in light of emerging technologies like synthetic biology, how do those relationships or governance structures need to change?
Thanks. That's a huge set of questions, but I'm glad we're highlighting them today. And building off of Rocco and Alexander's comments, right? Biotechnology can both pose risks, mitigate risks, essential to economic and national security. And so it's key to look at how we evolve our biosecurity and governance strategies as quickly as we are evolving those biotechnology capabilities. So you can think about biosecurity governance as broadly the set of processes that influence behaviors that allow us to prevent or deter misuse of biotechnology, as well as to enhance security. And there's many different strategies and there's many different organizations involved. And I think the bottom line is increasingly as our capabilities diversify and expand into all areas of our economy and ways of life, we need all these different organizations to be able to be working together, from private firms to government to academia. Right now, we can think about a range of both formal and informal mechanisms. In areas like safety, we have oversight policies and structures that range from individual groups at institutions all the way to the broad policies and the mandates that help govern that. In security, we have laws against the development or the use of biological weapons. We also have agreements internationally, right, that are prohibiting biological weapons development. And then increasingly, I think we don't put enough emphasis on the types of coupled research programs and informal social infrastructures that are needed to define the types of both technologies and behavioral innovations that are needed to develop better practices, better technologies, and then importantly, to share those lessons across organizations, both locally, across the country, and really across the world. And so, actually, my colleagues and I recently had a paper, I think it was now 2 weeks ago, in Science Magazine's Policy Forum that discussed some nascent pilots to try to get better at coordinating when really there is underdeveloped standards in risk management. So how do we just share what's working and what's not working over time? One of these is the International Genetically Engineered Machine Competition. This is a student competition in synthetic biology that actually involves more than just students these days. It involved over 4 countries, and it's convened a number of different organizations from all over the world that are testing their approaches to both promoting this technology and protecting the technology from misuse. And so that's everything from laboratory safety to these dual-use risk management strategies to personnel and again, social infrastructure. So I think the big lesson here is there are many different approaches. We likely need them all, and more coordination and research and systematic approaches would get us a long way.
Wonderful. Thank you. So I think your answer, Megan, and then Rocco's previous discussion of lab safety both sort of hinted at the need for risk governance strategies to sort of evolve alongside the technology. And so Rocco touched on that a bit on his discussion of lab safety and how should lab safety evolve in light of synthetic biology capabilities. Rocco, what I'd like to ask you about now is sort of building upon your discussion with lab safety. I would assume there's also a need to evolve controls in the sort of export control space or in the foreign investment space. And I know we got a lot of questions from our audience last week about both of those. So if you could talk to us a bit about what are the risks that we're trying to manage from the export control or foreign investment perspective, and whether you think those are sufficient for this evolving technological landscape.
Yeah, that's another really good question, because I think the entire community is struggling with what export control really means when you're not talking about established weapons systems. So, for instance, if you look at the underlying authority behind U.S. export control, you have the Wassenaar Agreement and the Australia Group, one of which, the Wassenaar, is more focused on real weapon systems, and the Australia Group controls are more focused on methods to implement the controls under the Biological Weapons Convention and the Chemical Weapons Convention to not export technologies that could be usable for the production of biological or chemical weapons. Neither one of them is focusing on the proliferation of technologies that aren't used explicitly for the production of pathogens or dangerous chemical agents. When you get into some of the risks that we discussed, like environmental risks, like potentially risks that might challenge notions of what people think is the definition of a human, animal rights risks, none of those regimes are designed to address those variety of risks. One of them that's talked about a lot lately is risk to the economy. Should export control regimes be modified to basically protect the U.S. economy, especially when a robust U.S. economy is needed to ensure that we have a robust military-industrial base going forward? So if we need to maintain hegemony over a particular biotechnology in order to have all of the cool gadgets and all of the cutting-edge materials to put into our next-generation military, should that be protected via export control? And I think that's a completely open question because, as I said, historically, export control really has been used in the context of the life sciences to control the proliferation of biological weapons and biological agents. I would say it's an active point of debate, and the Department of Commerce had a notice of proposed rulemaking and request for comment from the public that's out there on exactly what we should be thinking about related to foundational technologies and advanced technologies and how to regulate them. So I'd say if people have opinions about how that should go forward, someone's listening.
Wonderful. So you sort of highlight the challenge of the promote versus protect trade-off with the larger economy, and then specifically the bioeconomy, which I think is a great pivot again to discussing the promote aspect of the bioeconomy. So Alexander, getting back to that, investments in the bioeconomy, particularly investments that government agencies like the Department of Defense could be making. I think one of the challenges and opportunities in the synthetic biology space is there are many emerging technologies that are really the focus of the national security community right now, right? We talk a lot about AI and 5G. So I would like you to talk a little bit about, as we're thinking about how do we promote the bioeconomy, what trade-offs exist between investing in capabilities like AI and 5G? How does that affect our ability to invest in synthetic biology capabilities? And then what opportunities are there for synergies to benefit from investments made across sort of a portfolio of emerging technologies?
Yeah, that really is a great question, and it's of course not an easy question because the trade-offs. Everyone has a need, and there are lots of things that are very important to invest in. And so, I would say, first off, this is the first time in I believe ever that the Department of Defense has said at an enterprise level, when you're thinking of the top technology priorities for the entire department, it's in the top 10. So again, because and that is in recognition of we're now talking about what biotechnology can do both in the traditional biosecurity space, but then also in the much larger opportunity or application space. I would imagine now that it's even more preeminent how important it is given the challenges we have with COVID-19 right now. Defending, from the Department of Defense's standpoint, readiness and defending healthy, keeping their warfighters healthy is extremely important. And so that's both on the new types of biotechnology design, but then also the kind of technologies to help protect against something like an infectious disease. And then at the synergy level, if you think about anyone who has worked in synthetic biology knows that it's a lot of computer science. But for people who aren't working in this space, you might not realize that a huge amount of the work that goes into doing this kind of design, build, test of these new biological systems use computer algorithms. So what we consider AI, machine learning and deep learning, is central to how our ability to rapidly and at a really high scale and high throughput do these kind of analysis. So there's synergies across the board. If you think about, so the Department of Defense has 11 modernization priorities, and some of them AI, in biotech, obvious synergy, something like autonomy. So really what's happening with companies like Ginkgo and their peers for why synthetic biology is becoming such a technology tool is because of laboratory automation and using the robotics in the laboratory to get really high throughput experimentation. So autonomy in synthetic biology. So autonomy it goes much beyond just that, but there are fundamental overlaps between a lot of this. There's a lot of talk around DNA as a data storage medium, which is a synthetic biology application, which could have huge implications on the future of the microelectronics market, which is another modernization priority for the Department of Defense. So there are lots of overlaps. The trade-offs are not easy. I think that COVID-19 is showing us that it is truly important to be investing in this kind of technology, and I think that we'll only see more going forward.
Wonderful. So that, I think, also helps pivot to the next set of questions that I wanted to get our panel's thoughts on. I think we would be remiss to talk about anything biology related without specifically talking about COVID-19, which has shown that health security is a key and critical enabler of national security. So what I'd like to ask our panelists to do is help our audience understand how synthetic biology can help the world respond to the COVID-19 crisis, and looking forward, how some of these emerging capabilities might be able to help us prepare for pandemics in the future. So I'll start with Megan, and then we'll sort of go to each speaker. If you have specific examples that could help us and help our audience really get an understanding for what the technology is and how it could directly help us respond to some of the issues we're facing today, that would be great. So over to Megan.
Yeah, I think it's been really inspiring to see the synthetic biology and broader biotechnology and life science research community come together really all over the country and the globe to explore how their capabilities can be quickly developed in order to face this challenge. And it's really in a number of different areas. So we're seeing many different approaches in diagnostics, especially rapid point-of-care diagnostics, in the exploration of new types of therapies, as well as new types of vaccine development platforms, including ones that might be able to go beyond a single threat to be able to address many different types of threats in the future. One that's caught some attention and is interesting is Moderna's mRNA vaccines. There are other types of approaches in terms of diagnostics that are leveraging CRISPR and other types of technologies. There's been several synthetic biology sort of community gatherings to discuss some of this recently. Along those lines, we're also seeing many of the existing synthetic biology companies which have advanced platform technologies offer up those platforms for a diversity of different groups to be able to use their tools to test and scale. And so that has been an exciting development to see, to what extent are these platforms able to meet many different needs. And we're starting to see some of the pain points around leveraging those types of opportunities. But I think looking forward, some of the things around economic security and health security broadly is addressing these supply chain challenges that we're seeing around materials and what are the resources that we need rather than having kits. How do we address the long-term supply chains that we need and also rethink the requirements around certain types of testing and treatment. And so this relates to the last point I wanted to highlight. It's becoming clear that in all this, the coupling between our science and technology and the institutions and governance structures and broader economy is critical, and to have a fluid conversation around that when it comes to what types of regulation and safety standards might be appropriate, and how do we ensure that the strategies we're using today don't undermine our long-term security in moments of crisis. So I've been inspired. We've seen some pain points, and I think there's the opportunity to think beyond those to secure our nation and the world going forward.
Wonderful. Thank you. And Rocco, did you have anything to add particularly? I know Megan highlighted the supply chain issues. One of the things that I was going to ask the remaining panelists was to not only talk about opportunities to respond to COVID-19 and future pandemics, but what has our response thus far shown us about the insecurity, relative insecurity of our supply chains and where we might need to invest in the future. So, Rocco, if you have comments on that as well, I think we'd welcome them.
Yeah, I mean, I think one thing that's interesting is how we've already demonstrated synthetic biology can provide a global health security benefit. I think the astounding rapidity with which we identified the causative agent, had it sequenced, shared that sequence throughout the world, and then had samples basically shared throughout the world that people could begin to manipulate and test, is in no small part due to the same forces that created the tools that enabled modern synthetic biology, the ability, as you said at the kickoff, to rapidly read and then write and understand what a DNA, or in this case RNA sequence, means. I think because we've come so far on that capability since the 2003 SARS outbreak, we really were able to get a jump on at least the biochemical aspects of this outbreak, even though we seem to have dropped the ball on a lot of other aspects. I think another thing that's important, Megan mentioned vaccines. I think once we identify a vaccine candidate that is safe and effective, one of the ways that synthetic biology could really help get this outbreak under control is in advanced vaccine manufacturing techniques. And those are those that got away from growth of, let's say, an attenuated viral strain or a virus like particle and cell culture or eggs to something completely different. So let's say, manufacturing viral components in rapidly growing bacteria or in tobacco leaves that could be, transformed en masse to produce grams and grams quantities or kilogram quantities of this viral antigen extremely rapidly, or even completely cell-free systems, basically vats that have all the inner workings of cells so can produce virus components or vaccine components very easily, but don't have any limitations of needing to grow up the cellular factories that pump that material out. So I think some of the work that's been pioneered to kind of break some of the trade-offs we see every year with a seasonal flu vaccine in synthetic biology could be leveraged to reduce the time from which that we have a vaccine that is safe and effective to the time where we're getting it into the billions of people's arms that need it. and I'm looking forward to seeing how that benefits.
Great, thanks. Alexander, anything that you wanted to add to those two comments?
Yeah, absolutely. So really what we're describing here is biomanufacturing and the promise that biomanufacturing provides, both on the vaccine space, but then to we were talking about before, things that are outside of the medical application area as well. So if you're talking about supply chain security, when international travel is restricted, borders are closed, and we need to be able to produce other things, commodity chemicals and other types of critical commodities that we need in the U.S. or in whatever country you're in, biomanufacturing really has the potential to provide that kind of solution. So I know the Department of Defense is investing in this. They've already announced that they're going to establish a Bioindustrial Manufacturing Innovation Institute, which is an investment in the U.S. capability to manufacture using biomanufacturing techniques, synthetic biology and engineered biology techniques to do that. And then the other thing I would say is, so to your earlier question about what are the risks of the federal government not investing, a lot of the partners, synthetic biology companies that are the partners in this response have been strong partners of the Department of Defense and have been research partners and recipients of DARPA awards and other types of funding where there's been those longstanding relationships and investments from the DoD side into these technologies. And now we're seeing these companies be able to both have products, but also step up and respond in the event of a national security crisis like we're seeing with the pandemic right now. So it is a great example of why of the success that we've been investing in over the last 5 to 10 years from the Department of Defense's standpoint, and then why we need to continue to do so going forward.
All right, so we've got about 10 minutes left, so I want to make sure we get to some of the great questions that have been submitted both this week and last week. One of the questions I'll pose to Megan was really asking about unclassified research, particularly in academia, in synthetic biology, and what protections are available for that type of research that's being done in the unclassified, open academic environment.
I think it's important to think about what we mean by, protections in this space, and also what is our overall strategy for thinking about what will enable us to have, economic and national security. And time again, we've come back to the importance of being able to, share information and our results, because a lot of the challenges here require scientific collaboration, and they require us to be able to work, fluidly together across the globe. And this question of whether or not there are categories of information that are sensitive and what to do about them are really at the heart of some of the discussions recently around dual use. But that being said, if we think about our strategy for what we need to do in order to ensure that we are capitalizing upon our investments in research, then we need to think about the broader ecosystem of research. So how are we making sure that we have the translational capacity coupled to our investments so that we can build out and maintain, right, the types of companies and innovations here in the U.S., and so that we can also recruit and retain the talent when we have the capital around that. And so it's not just a protection, it's how quickly are we able to actually, capitalize upon the investments there. That being said, I think that there's another need that's not being addressed, which is thinking about the infrastructure choices around planning in biotechnology going forward, and how investments like the biomanufacturing institutes that Alexander highlighted can bias the overall vulnerabilities and governance opportunities in the field. So if we are looking at more centralized or more decentralized capabilities, these have trade-offs both in terms of efficiency and our ability to secure information and materials within that. And so a lot of those types of questions and really strategic questions, I think, are understudied and underaddressed as we began to make these large-scale investments. And so I think about, we really need to diversify our thinking about what protection and security means in light of how our choices and investments in technology will affect our ability to protect them and be secure in the future.
Great, thank you. So I'm going to try to combine a couple of questions that were submitted at the last session, and I'll pose them to you, Rocco. I think our audience members are trying to get a better understanding of where U.S. capabilities, where existing capabilities within the U.S. compare to other countries or non-state actors, and additionally how biological weapons compare to other types of weapons. The questioner was asking, what is the risk posed by biological weapons versus nuclear weapons? So if you could give us sort of a brief understanding of where is the U.S. in this technology space relative to some of adversary nations or non-state actors, and how does the risk compare to other weapons we might be more familiar with?
Okay. Well, addressing the second part of the question first, related to the comparative risk of biological weapons and biological agents, let's say, made with synthetic biology versus others, I would say that no one knows for sure. However, we do know that the capability to cause harm on a scale equivalent to nuclear weapons has existed for many, many decades, since the late 50s and early 60s, when in the defunct U.S. offensive biological weapon program, basically pioneered weapon systems to use pathogens to infect potentially millions of people. And then that was copied and also independently invented by other countries. But that all used technology way before synthetic biology or even recombinant genetics was a thing. So really the question is, what could synthetic biology do to make biological weapons that much worse? And the answer is, if you think about it, there's a lot that could be done with technology that happened, that existed for many decades, that wasn't chosen to be done, that could make biological agents a lot worse. One obvious one relates to antibiotic resistance. We've known how to make bacteria resistant to multiple antibiotics and how to retain their fitness and pathogenicity for many, many decades. Doesn't rely on synthetic biology, and yet if you have a biological weapon that can be disseminated and also is resistant to the treatments that we use against it, that would be incredibly potent. The other thing to keep in mind is, as we said at the outset here, the vast majority of synthetic biology research has nothing to do with pathogens. So the things that would cause harm, that have been leveraged to intentionally cause harm, are not really on the table for the vast majority of researchers in synthetic biology. That being said, one of the interesting thrusts of synthetic biology research is in the first part of the design, build, test cycle, having a greater understanding of the rules that underpin how biochemistry works, so that someone who wants a biological product with a particular function can use, let's say, a software suite to just design that using real terms. Like, I want a protein that binds this cell type and then delivers this function to that cell type. When we have that ability to use, just say, common language to get to a engineered biological function, then that unlocks a whole bunch of beneficial potential, but it also unlocks some nefarious potential in that people could basically begin to design toxins or organisms decorated with those molecules to have effects, deleterious biological effects we've never seen before. And because those would be difficult to predict a priori and make a list of things that should be controlled, it would be extremely difficult to fit inside our export control regimes or even the controls that we have within the country, because they wouldn't fall within a list of proscribed pathogens. I don't know, in lieu of time, if you want to answer the first part, I should skip. But
that was a great, I think, landscape of the broader risks associated, and I think that gets at what the questioner was asking. we are running out of time. I do want to end with one question that did come in from the audience, and I'll pose it to Alexander. There's a question about strategy development. And this ties back to some of the discussion we had with Dr. O'Toole last week. She really highlighted that China sort of has a top-down strategy for many technologies, biotechnology in particular. And, I think going forward in the post-pandemic world, the national security community is probably going to rethink its broader biosecurity strategy to include emerging technologies like synthetic biology. So how does one go about thinking about the future biosecurity strategy, and how should we incorporate emerging technologies like synthetic biology? And that's to Alexander. Yeah. Sorry, also a hard question.
But that is a great question, and actually something I wanted to follow up on Rocco's commentary as well. So when you think about this, it gets overwhelming if you put it in the context of, you can't predict what's going to happen, you can't quantify, you can't calculate, you can't know what all the potential risks are. But when you think about an infinite threat space, if you will, the way to mitigate an infinite threat space is to be world leading in characterizing, developing, and producing countermeasures to those kind of threats. And so this gets exactly to your question of how do we have a strategy? And so the strategy really needs to be, and it's easier said than done, the global leader in producing and managing and manipulating and developing technologies out of synthetic biology. And there's always a lot of comparison between countries that have the Made in China 2025 and all these kind of top-down strategies, but the U.S. is by no means the world leader in controlling information and top-down mandate of anything. We are the global leader in bottom-up innovation. But then if you see the times throughout history where we have then taken bottom-up innovation and put it towards a purpose, you take the race to the moon, right? We had a phenomenal aerospace innovation in our academic sector, and then all of a sudden we were racing to the moon as a country, and we focused on a singular outcome that was not top-down mandated. It was challenged. It was a challenge problem. If you think about the same thing with the Human Genome Project, we had phenomenal scientists across the country and collaborators across the globe, and then we had a focal challenge problem, and we sequenced the human genome. And so the strategy really needs to be to continue to have this phenomenal innovation base that percolates up, and then in times of need, we need a well-coordinated response there. That's really where our challenge is right now, is coordination, because there's so much going on. There are so many people who want to help, so many companies, so many academics who have phenomenal ideas, but there's not a coordinated way to ingest all of those ideas and put it into a kind of productive momentum forward. And so as we think of a strategy, that coordination strategy where we're still investing in the components, a ton of the different components across the country in terms of applications for this chemical or this material or this vaccine, and then being able to pull those resources together and coordinate towards a common goal like a COVID-19 response or COVID XX response is really where we need to go with those strategies.
Well, wonderful. That was a great forward-looking way to end our conversation today. And I want to close by, again, thanking all of our panelists for joining us to discuss this really relevant and important topic. I also want to remind everyone who's watching online, this particular event is part of a larger series about synthetic biology at CSIS. So please stay tuned for future events where we will continue this conversation. Thanks for tuning in, and thank you again to our panelists.